the sound of what Peter Lansdorp does, cell immortality. That's very encouraging. Okay.
It really is a thrill to be here. I like to go to sessions in biomedical meetings that I usually attend, and I go to areas which are not really my area, and the way I hear talks that are which are not really in my particular line of field, and I feel like I take home more than if I stay within the narrow confines of my particular specialty. And this whole meeting has been an exaggeration of that. In fact, it's like the mecca of lateral thinking, and I really think I want to thank the organizers for allowing me to be here and to present some of this stuff. I also want to make a comment. I mean, my roots are in the old world. I wasn't born in Canada, like several or many of you, I do believe. And I chose to live in Canada, not for some of the things that were mentioned by N. Golden, that there's a lot of things going for Canada, and that they just have to do with the quality of life, the space, and the fact that a lot of things in terms of public policy have been done right. Of course, there's now discussions, things that could be done better, but there's still a lot going for it. And I could live in Menlo Park. I mean, I've certainly been offered several times, but I prefer to live in Canada. In fact, I could live in Toronto. I mean, throw in the scenery, I would probably be here because you have more of a critical mass here. And one of the things that is actually telling is throwing a meeting like this here. I think it's really been spectacular. And once again, thanks for the organizers. Okay, so I'm going to talk about recent developments in molecular biology and in cloning and in telomeres. And they have to do with mortality and immortality at the level of biology. And for those of you who get scared about these issues or who really are not into biology, I'll give you the take-home message. And the take-home message is that at the cellular level, in biology at least, mortality is programmed, and immortality is also programmed. And we're getting a pretty good understanding as to the molecular machinery that are involved in these programs. So the story begins with Dolly, the famous cloned sheep. So just a few years ago, things are going really fast. We had this report that it was possible to take a cell from an adult animal and clone an animal. So just to remind you, before 97, it was known that you can take an early stage embryo, so before implantation in the womb, and disaggregate the embryo and take the cells, let's say at the 100 cell stage, and basically take the nucleus from such a cell and transfer it into an egg from which the nucleus was removed, so an enucleated egg. And then by this type of nuclear transfer, you can now start the process of development and create an organism, because the genetic material, of course, at this stage is present in all the cells. Well, then by Dolly, it was shown that you can actually take cells from adult tissue. In fact, these were cells from mammary glands, so the cells that produce milk from an adult sheep. And one can take a nucleus there and do the same thing. And this was, of course, a very important experiment because it showed a few things. First of all, it showed that the genetic material in all the cells of our tissues, at least in the case of the sheep, but there's now no reason to assume that it's not the same in humans, is preserved and can be used to recreate a complete organism. This is important because there's still a lot of theories about aging which deal with the accumulation of mutations, and just the fact that you can recreate this here indicates that at least not all the cells are subject to mutations where in fact life would be compromised. And secondly, it has therapeutic applications. And these therapeutic applications have to do with stem cells, which happen to be my area of interest. So I'm a cell biologist looking at blood-forming stem cells. But the realization that you could clone sheep and cows and mice, a lot of animals have been cloned since, has implications for a strategy which is going to have therapeutic potential which is called therapeutic cloning. The idea is here not to clone a human being just to have another kidney or another heart. The idea is to create stem cells. So you would take the nucleus from a patient, any type of tissue. In fact, you can take those cells and engineer them so a defect might be corrected, and then take that nucleus from that individual and put it in an enucleated egg. And of course, there's issues, ethical issues, related to the origin of those eggs. But in principle, this is possible, and I mean, there are a lot of technology is directed towards some of these issues. One thought that crossed my mind at this meeting, which might be a direct result of it, if it were only possible to, say, to recreate the oocyte, so to create artificial eggs. So I thought maybe we should start a company, artificialegg.com or something like that, because then we would get around all these issues of cloning, because then we could do the same thing, start embryogenesis, and then this is all done in the laboratory, take this embryo before it would normally implant. That's where we would stop, and that's where we draw the line. We could disaggregate the cells and create what are called embryonic stem cells, and we know that these cells can be pushed to differentiate into various lineages, and this is a very active research area. We know from studies in the mouse, for example, that you can take these embryonic stem cells and push them to differentiate into, for example, insulin-producing pancreatic island cells. Well, just on the way flying in here, I read an article that in Alberta, people had shown that you can treat diabetic patients by transplanting island cells, so the cells that produce insulin, and thereby these people no longer have the need to inject themselves with insulin on a daily basis. So there's a lot of excitement about that, but that approach really is the classical transplantation strategy. Whether you transplant island cells or kidneys or hearts, livers, you have tremendous problems with the immune system, which is tuned to recognize what's foreign relative to self and to deal with it in a very aggressive and destructive way. So all transplant patients are immunosuppressed. They have to take drugs to keep their immune system at bay. And in fact, the major contributors on the waiting list for, say, kidney transplant are the people who had a kidney before. Well, if you could do this with a diabetic patient, cells from a diabetic patient, you could essentially create cells which would be genetically compatible, and there should be a true cure then for that. Well, I can go through these other tissues. It's not really the main focus of my talk, and I'm really not involved in this directly. I'm interested in these stem cells issues and issues related to mortality and immortality, which have to do also with cancer. Okay, so then from the same group that cloned Dolly came the story 2 years ago, or 1 year ago actually, that there was a problem with Dolly. And it was mentioned that, in fact, Dolly was a sheep in lamb's clothing, because although obviously DNA had been used to reprogram and recreate an animal, there was something wrong with Dolly, and the problem had to do with the ends of chromosomes called telomeres. Okay, well, that happens to be where I'm very interested in. And for that, I have to take you one step back to, and this is in fact my design slide. Okay, so there's genetic material can be packaged in 2 ways, and a very simple design is to do it circular. This is probably the most common way whereby genetic material is packaged and it's transmitted. And for example, E. coli, and I have no problem using the abbreviation, packages DNA in a circle. However, if you want to have very large genomes, and DNA is a helical molecule which forms coils and twists, using this form of packaging has its limitations in terms of the amount of DNA that you can package. So an alternative would be to use a single line. In fact, that has been spectacularly successful, because that has allowed also the evolvement of diploid genomes, where in fact every cell has 2 genomes. And this has allowed a rapid evolution, because now traits, genes of encoding properties could be combined, and you get a thing like sexual revolution, really, because this has allowed tremendous expansion of diversity. The bottom line here is that if, say, with a single copy or a circular piece of DNA, if you're doing just fine, well, you're doing well. It's not going to be good enough if you can cross with properties that give you an advantage. Natural selection would deal with you, and natural selection is clearly favoring sexual reproduction. The only problem with these linear chromosomes is that you have to deal with a very basic aspect of cell biology and molecular biology, and it has to do with the mechanisms of replication. So all DNA polymerases, so if we now look at here, we consider this the end of a chromosome. DNA polymerases are unidirectional. That means that they can only replicate DNA in one direction. So typically, you talk about a leading strand and a lagging strand. And the lagging strand basically is always trying to catch up with the leading strand. And the lagging strand synthesis has a problem for the very end in that it has to start with a labile primer. I don't want to get into too much detail, but the bottom line is that at the end of a replication line round, inevitably you're going to have a piece at the end of a chromosome that is missing something. So if you want to have your DNA or your genetic material in linear chromosomes, you have to solve this problem. Well, the problem to this solution is very old. In fact, it's shared by plants, worms, and humans. And the solution is an enzyme called telomerase. And essentially what this enzyme does, it takes an RNA template to encode the DNA and basically add pieces which are lost with every replication round to the 3' end of chromosomes. So this enzyme is clearly essential then to prevent this shortening of chromosomes, and in fact, it is essential for life, as I'll explain to you. So I mentioned that this is a very old machinery. I mean, there are some organisms, such as Drosophila, or fruit flies, which seem to have lost this particular enzyme, but as I mentioned, so plants, worms, and humans all use this particular enzyme, and in fact, the template, so the RNA that is used to make DNA here, the sequence is very similar between all these life forms. And for example, in plants, the caps of chromosomes are characterized by repeats, so hundreds or thousands of repeats of this particular structure, again encoded by this enzyme. Okay, so I was really very interested in this telomere shortening very early on because I found it in blood-forming stem cells, where we were thinking about cell renewal potential, and we were looking for protein factors that would allow expansion of stem cells for bone marrow transplantation and for gene therapy. We found that the telomeres got shorter, and but the problem was that you need these techniques to measure telomere length were laborious, so I put a lot of time and effort into developing new techniques to measure telomere length, and I was fortunate that people in Denmark had come up with a new type of synthetic molecule, which is really a hybrid between a protein and a DNA oligonucleotide, which has unusual properties, which essentially allow, if you now put a tag on such a molecule, a fluorescent tag, to measure the length of telomeres. So essentially what you do, you would break open the DNA double strand, which is held together by hydrogen bonds. You can do that by heat and solutions such as formamide. So you denature DNA, and then you hybridize with this probe under conditions that do not allow renaturation of the strands. And sure enough, you end up with fluorescent tags, fluorescent molecules at the ends of chromosomes, which relate to the amount of the repeats that there actually are. And I cannot describe what I saw when I first looked under a microscope using these probes, and after having figured out how to do it. In fact, I'm mostly reminded by the aesthetic beauty of this stage here, because it really is spectacular to just look at human chromosomes, for example. Might make it a little darker if that's possible. And because what I saw was very encouraging, in that now every end of a chromosome lighted up, and the amount of fluorescence was very similar between the two sister chromatids. These are what are called metaphase chromosomes. These are genetic material after duplication, and they are again templated on one another. And you see either very faint spots or you see very bright spots, which indicate that indeed this fluorescence was quantitative. And that was really that's a novel concept in situ hybridization, because with regular probes, there always is this competition between renaturation. Anyway, oops, pressed the wrong button, I guess. So, take-home message is, telomerase was found to be expressed in immortal cells and in cancer cells, but not in most normal cells. So with each cell division of our tissues, telomeres get shorter. And of course, this is not happening in the cells that are forming the reproductive tissue. So that allows me to go back to a very old question. What came first, chicken or the egg? And the answer really is that there was a very primordial cell which evolved through this mechanism of sexual reproduction to give rise to a carrier, which would then basically transmit and shuttle in time, using these mortal carriers, which are chicken and which are us, to survive and to have a selective advantage. So, what about the question of our own mortality? I mean, do these insights? So we now know that if you, for example, delete the gene, and I've been involved in those studies, delete the gene either for the template of telomerase or the actual reverse transcriptase, you get a very similar phenotype in mice. You can do these kind of studies using, again, these embryonic stem cells in mice. And what you see is that mice become infertile, and the number of generations that it takes before they become infertile is strictly dictated by the number of these repeats that you see before you start the experiment. Now, mice happen to have very long telomeres, and humans happen to have much shorter telomeres. So the question then was asked by Isabel Hoffman, can we achieve or will we achieve genetic immortality? And I think the answer is, well, it's really now, it's been done because we've recently shown that if you take cells which are very old and which have short telomeres, and in this case it was done in cattle. So, and you can see differences as in cattle, as you can see in humans, as you can see in other species, baboons, for example, we looked at. You take very young cells from young animals, they have typically a lot of these repeats at the ends of their chromosomes. And if you look at very old cells, they have very short telomeres. In fact, this is a mechanism whereby growth is also limited. So it's a way to prevent unlimited growth. And in this sense, it has a role in protecting against cancer. But it also has the problem of encouraging genetic instability, because if chromosomes are uncapped, they do not have sufficient of these repeats, they have a tendency to fuse, and then you get structures which may break up and may allow you a loss of genetic material, and that's something that cancer cells really like. So these things are important. Anyway, for the cloning that was subsequently done with cow cells, we took cells, and this was in collaboration with a company in Massachusetts, Advanced Cell Technology, so Bob Lanza and his colleagues there. They took cells from a cow, allowed them to grow till they didn't want to grow anymore, and then we took the nucleus from those cells and basically created cows. In fact, 5 from a population of cells, but in principle you can make thousands of genetically identical animals. And what we found is looking at the telomere length using these techniques, in fact that there was nothing wrong. In fact, if anything, the telomeres in these cloned animals were longer than what we saw in the age-matched controls. So that clearly makes the point, and I have no doubt that that you can take a cell from these cloned animals and do that in perpetuity. So, because genetic material would be maintained, you essentially have maintained at least genetic immortality. So we've answered at least one question at this meeting. The point, of course, is, well, where is this all going to go? I mean, I think these kind of studies are important because they're telling us fundamental things about mortality, and as I mentioned, this relates to how tumor cells operate and how we might be able to interfere with how they operate. And of course, we have to consider in the end the question, when is enough enough? And I suspect it's going to be difficult to deal with this telomere shortening in the cells of our bodies, because it's going to be difficult to allow for the kind of changes, telomerase, for example, expression in all cells. And in fact, it may be more important in a cell from the kidney from one individual, and in another cell of the blood vessels may be more limiting than in lifespan in others. So I want to end with the question, which I think is very appropriate for an end, that is, when is enough enough? And I think that kind of question we will have to face much more directly at a personal level in the future, because we will be able, we will have these tools to maybe prevent cancer or cure cancer, maybe extend lifespan by transplanting at least some primary lifespan limiting organ failure. But at one point, we will have to, and that's something we really have to come to grips with, thinking now about the longer future. When is enough enough? So I want to stop there. Thank you.